Okay, let's get into this. We are going deep into the soil today.
Right into the plant tissues, even.
Exactly. We're looking at how scientists are, well, essentially building these custom microscopic defense systems for crops. The goal is to replace chemical pesticides and, boost plant health.
And we're going to unpack a really fascinating study on this from horticulture research.
Right, from the Chinese Academy of Sciences. It's all about something they call engineered synthetic microbial communities, or SynComs for short.
What's so amazing here is that it's really getting back to nature's own blueprint. The whole idea is based on plant endophytes.
And those are the microbes that live inside the plant, right, symbiotically.
Exactly. They're crucial for everything from, helping the plant get nutrients to fighting off disease. SynComs are just the custom-designed versions of these teams.
But I imagine it's not so easy to just build one of these teams and have it work.
Not at all. That's been the central challenge for a long time. How do you assemble a group of microbes so that they're stable, they actually function, and the plant accepts them?
Okay, so the researchers here, they focused on edible lilies grown in a monoculture.
Continuous cropping. Year after year, same plant, same soil.
Which is a famously stressful situation for soil, right? It usually makes plants more vulnerable to disease. So what did they find going on under the surface?
Well, this is the interesting part. They found that the stress didn't just weaken the system. It actually triggered this kind of intense microbial arms race.
An arms race.
Yeah, it created what they called an antagonistic equilibrium. So yes, the soil-borne pathogen, Fusarium oxysporum, it was there and enriched, but, and this is the key, so were the beneficial defensive bacteria like Pseudomonas and Bacillus.
So the plant itself is somehow encouraging its own bodyguards to show up to the fight.
It's actively selecting them. The data showed that specific families like Burkholderiaceae and Pseudomonas were the key players maintaining this balance.
Okay, and this is where it gets really specific, I thought. The part about where the microbes came from.
This was the jaw-dropper. They found that about half, so 50% of the endophytic bacteria came from the surrounding soil.
Right.
But less than 10% of the fungi did.
10%? That's a huge difference.
A massive difference. It shows this incredibly strong, very specific selection pressure the plant is putting on its fungal partners. It's extremely picky about which fungi it lets in.
So that pickiness basically gives the researchers a map. They can see who the plant wants on its defense team.
Precisely. They isolated those core players, bacteria like Rhizobium and Methylobacterium and the real star, a fungus called Telleromyces. Then they started building different SynComs to test.
And what was the biggest practical takeaway when they put them to work?
The first one was just pure synergy. The multi-strain groups, the consortia, they consistently and substantially outperformed any single microbe on its own.
In both promoting growth and fighting the pathogen.
In both. The community is always stronger.
Wait, so if the community is what matters, was there a secret ingredient, a specific type of microbe that made the real difference?
They absolutely found one, and this kind of flips the script on how we've been thinking about this. The SynComs that contained fungi, specifically that Telleromyces strain, were significantly more effective.
More effective than the ones with just bacteria.
Way more effective. It just confirms that this isn't a one-kingdom job. You need that complexity. You need the fungi and bacteria working together. We can't just rely on the usual bacterial suspects.
I mean, that's not just some small lab result. That offers a real sustainable solution for huge agricultural problems like replanting challenges or Fusarium wilt.
It's a clear strategy for green agriculture, for cutting down on all that pesticide overuse.
We've moved past just, hoping the right microbes are in the soil.
Exactly. Now we can rationally design and deliver the precise team that we know will work. It's a beautiful bridge between microbial ecology and day-to-day farming.
It really is. And it all leaves us with this final thought for you to chew on. The researchers found this antagonistic equilibrium network because they looked in a stress system, right? In that continuous cropping field. So what other unexpected microbial balancing acts are already happening? Right now, right beneath our feet, in other farming systems that we could be harnessing? What other specialized fungal partners or plants out there desperately trying to recruit that we just haven't even met yet?